The overall goal of this procedure is to observe and analyze the effect of cell-matrix interactions on fiber realignment and matrix remodeling activities in a fibrin gel system. This is accomplished by first polymerizing a three-dimensional fibrin gel containing microbeads onto cover glass contained within an environmentally controlled bioreactor. The second step is to prepare a highly concentrated cell suspension that is used to position clusters of fibroblasts, or explants, into geometric patterns on the surface of the gel. Next, the sample is imaged every 15 minutes using differential interference contrast microscopy over the course of 48 hours. The final step is to measure the extent of fibrin reorganization and remodeling in the form of strain from different regions of the gel using digital image correlation. Ultimately, this explant system allows one to arrange the cells into simple geometric patterns, which makes it easier to visualize and probe the mechanisms underlying cell-driven matrix remodeling and fiber realignment.
This method can answer key questions in the field of mechanobiology, such as what are the effects of varying spatial and temporal mechanical cues on cell behavior and the resulting changes these generate in the surrounding tissue matrix. Demonstrating this procedure will be Aribet de Jesús, a graduate student from my lab.
Gently dissolve fibrinogen into pre-warmed saline at 37 degrees Celsius overnight to create a 33.3 mg per milliliter stock solution. Prepare a stencil on parafilm to lay out the location.